Continuation. Case selection. Fairly constant or predictable, more certain environment. Density dependent mortality, type 1 and type 2 survivorship. Type 1 means human, type 2 means mice, etc. Type 3, that means marine invertebrates, comes under R selection. A greater amount of death rate during initial stages or during infant stage, early stage death in case of type 3. That is R selection. Here, type 1, that means there are more adults which are dying than infants. So, type 1 human, type 2 most probably mice, deer, etc. Yeah, a constant rate of death over lifetime. Population size, constant in times, near carrying capacity, slow development, selection which is slow development, low rate of increase, delayed reproduction, large body size, heteroparity, repeated reproduction, few larger offspring, high level of social organization, high usually more than a year lifespan, successional stage, late climax stages, leads to high efficiency. Population regulation. Population can be regulated by density dependent and density independent factors. Density dependent factor are those whose effect on the birth rate or death rate changes as a function of the population density as population size increases, either birth rate decline or mortality rate increases or both. It is a negative feedback. Are those density dependent factor are those whose effect on the birth rate or death rate changes as a function of the population density as population size increases, either birth rate decline or mortality rate decrease or both. Density dependent factor are those whose effect on the birth rate or death rate change as a function of the population density as population size increase, either birth rate decline or mortality rate increases or both. It is a negative feedback. Not always density dependent factors are negative related to population size. In some case, growth rate increases the population size. And density independent factor are usually associated with abiotic factor, change in the physical environment that either promote or repress population growth, but their effects are independent of population density. Metapopulation, a set of local population connected by dispersing individuals, is known as a metapopulation. Metapopulation is a regional population composed of many local population, each of which may be extant or extinct at any level of any one point at time. It is an important concept for understanding population dynamics. PG habitats, where the rate of migration into and out of the subpopulation is great. Change within the subpopulation mirror those of metapopulation. Relatively small isolated subpopulation may suffer local extinction, especially if the habitat patch is small. Ecological characters. Community 1, Community 2. Dominant species. All species are not equally important, but there are only a few overtopping species, which by their no and growth modify the number and growth modify the habitat and control the growth of other species of the community. Those species are called dominant species. The dominant species are highly successful in a particular habitat. Keystone species. There are species upon whom several species depend and whose removal would lead to a collapse of the structure and ultimate disappearance of those other species. Such species are referred to as keystone species. Then species diversity. A community can be characterized by its diversity, which is a function of no different species it contains and their abundance. Diversity depends on species richness, the total number of species, and on the evenness, equality of the abundance. Community 1, high effect, evenness and low dominance. Community 1, high evenness and low dominance. Because all these three species, each species is of 25%, but in case of next one, 70% of as one species, is that there is a great amount of dominance, high dominance and low evenness. Community 1 and community 2 have the same species richness, but they have different species evenness. Why same species richness? Because there are 4 different species, that's why. Calculate biodiversity, diversity indices. Simpson's diversity index. Biologists use a more complicated formula called Simpson's diversity index, which calculates biodiversity rather than simply population size. D is equal to n into n minus 1 by small letter n into n minus 1, where D is diversity index, simply a number with no units. Always remember, no units. Okay. And total number of organisms of all species found. Total number of individuals found in the species you are interested in. That is small n. Diversity index, Simpson's index. Simpson index measure the probability that 2 individuals randomly selected from a sample will be belonging to a same species. Simpson index measure the probability that 2 individuals randomly selected from a sample will be belonging to a same species. A probability is analyzed here. D is equal to n by small n by capital N whole square, or D is equal to n into n minus 1 by n into n minus 1, where n is total number of organisms of a particular species. Yeah, here signs are kind of reversed. In case of earlier one, these are both same thing. Okay, capital N means here total number of organisms of all species found. The value of D ranges from wait a minute, I got confused. Yes, there are 2 things. One is Simpson index. Next is Simpson's diversity index. Okay, so first one we have here, don't look at this, you look at this. That is Simpson index D. D. Simpson index D. What is Simpson index? Simpson index measure the probability that 2 individuals randomly selected from a sample will be belonging to a same species. So capital D is equal to small n by capital N whole square, or D is equal to summation of small n into n minus 1 by n into capital N into n minus 1. Whereas, wait a minute, where small n is equal to the total number of organism of a particular species, which is very important, which is the total number of organisms of a particular species. Capital N, total number of organisms of all species. The value of D ranges from 0 and 1. With this index, 1 represents the infinite diversity and 0 no diversity. That is Simpson's index. Next is Simpson's diversity index. The whole scenario changes. 1 minus this D. 1 minus D. That is Simpson's diversity index. This is a measure of diversity which takes into account the number of species present as well as the relative abundance of each species. As species richness and evenness increase, so diversity increases. Shannon Diversity Index. Shannon Diversity Index. Diversity indices are statistics used to summarize the diversity of a population in which each member belongs to a unique group. For example, in ecology, the groups are typically species. In ecology, species richness refers to number of species, and species evenness refers to homogeneity of species. That is, the more equal the proportions for each of the groups, the more homogeneous or even they are. Different fields of application may be using different terminology for this concepts. PI log PI, Shannon Diversity Index. Shannon Index is an information statistics. The assumption is that all species are presented in a sample and that they are randomly sampled. Shannon Index H is equal to summation PI log PI. P is the proportion n by n. N is the particular species found in a population n. Capital N. In is a natural logarithm. Summation is sum. This is the number of species. Sorensen's coefficient, community similarity. Sorensen's coefficient of community based on the species present or absent. Number of species common for both the communities. Number of species of community 1, number of species of community 2. CC is equal to Sorensen's coefficient is equal to community similarity is equal to 2C by S1 plus S2. Disturbance and species diversity. Biodiversity is significantly affected by disturbance. Species diversity has been found low at low intensity of disturbance because of competitive exclusion. In such case, competitively dominant species exclude competitively inferior species. Similarly, at high intensity of disturbance, species diversity has been found low because only good colonizer or highly tolerant species can persist. Then both theoretically and experimentally, it has been demonstrated that species diversity is high at intermediate intensity of disturbance due to mix of good colonizer and good competitive species. Intermediate disturbance hypothesis. Island biogeography. Large island may ultimately have a larger equilibrium number of species. Small islands than small islands because immigration rates tend to be higher and extinction rates lower on large islands. New islands tend to be having a larger equilibrium number of species than for island. Nearer island tend to be having a larger equilibrium number of species than for island because immigration rates to near island higher and extinction rates lower. Island biogeography theory states that the number of species on an island is determined by the immigration rates or new species. Immigration rates of new species to the island and extinction rates of species on the island. Immigration rates to the island decline as its distance from the mainland increases. So here this graph is very important. Species interaction. This table is used for a quick recap of this all population interactions. Species A benefits, species B is also getting benefited, so mutualism. Species A is getting negatively affected, species B is also negatively affected, competition. Species A is positively affected, species B is negatively affected, predation or parasitism, same case. Then species A is getting benefited, species B is getting not benefited, not at all, affected in a negative way, so no effect. That means commensalism, but species A is negatively affected, but species B is neither benefited nor harmed. It's amensalism. Ecological independence and interaction. Here, there are different types. Proto-cooperation is also a beneficial interaction for both. First is neutralism, that means no interaction. Competition, direct interference type. Competition, resource use type. Mensalism, parasitism, predation including herbivory. Predation, commensalism, protocooperation and mutualism. Protocooperation, interaction favorably to both but not obligatory. That is protocooperation. Three major type of symbiotic relationship is both organisms benefited in case of mutualism. Lichens are formed by the association of algae and fungi. Nitrogen-fixing bacteria, rhizobium. Termites feed on wood, but they do not possess cellulose digesting enzymes. Flagellates such as Trichonympha lives in the alimentary canal, help in the digestion of cellulose. Coral reefs are formed by the association of cylinderates. Mycorrhiza attaches to the roots of plant like pine. In ruminant mammals, flagellates and bacteria assist in their cellulose digestion. These are mutualism. Then comes competition. Competition occurs when two individual species fight for resources such as food, space, light, water, shelter, etc. Interspecific competition is there. Interspecific competition between species. Interspecific competition within species. Intra-specific competition. Interspecific competition, here we can see tiger and bear. Intra-specific competition, two lions. They both compete for mates or for space or for territory, whatever it is. Biological interaction is the effect that a pair of organisms living together in a community live on each other. Competition occurs when two species have same resources such as food, space, light, water, shelter, etc. Competitive exclusion, ecological separation of closely related species when they occupy the same niche. Outcome of competitive exclusion, no species will be even. Niche differentiation will be occurring, local extinction of species. Competitive exclusion principle, when two species occupy the same niche or coexist the same ecosystem, then one will be excluded from the community due to intense competition. Two species cannot exist or coexist in a same niche. Competitive exclusion. Squirrels, Paramecia aurelia, Paramecia caudatum. They just, one of them extinct. Separate cultures both thrive, but when they are combined in a culture, Paramecium caudatum declines. Their number declines. Okay. Next, resource partitioning, a consequence of cost competitive exclusion principle. When two or more species divide out a resource such as food, escape space, shelter, nesting site, etc., then resource partitioning occurs. Resource partitioning avoids competition. Realized niche divides the resource among several species. That is resource partitioning. Character displacement. Character displacement is described as an evolutionary change that occurs when two similar species inhabit the same environment as a result of resource partitioning. For example, two closely related species of Galapagos finches, a significant difference in beak depth has occurred. Resource partitioning avoids competition. Lotka-Volterra model assumption. This is very, very, very important, extremely important assumption. The prey population will grow exponentially when the predator is absent. That's right. The predator, no, up to a limit. Okay, if the source is limiting, then the prey population will decline. That's there in reality. But the prey population, in case of Lotka-Volterra model, the prey population will grow exponentially when the predator is absent. The predator population will starve in the absence of the prey population as opposed to switching to other type of prey. The prey population will grow exponentially when the predator is absent. The predator population will starve in the absence of the prey population as opposed to switching to another type of prey. predators can consume infinite quantities of prey. There is no environmental complexity. In other words, both populations are moving randomly through a homogeneous environment. These are not reality, but just for analysis. Just remember this. Okay. dn by dt is equal to Rn k1 n minus n1 minus alpha 1 to n2 by k1. dn1 by dt is equal to r1n1 into k1 minus n1 minus alpha 1 to n2 by k1. dn2 by dt is equal to r2n2 into k2 minus k2 minus n2 minus alpha 2 1 n1 by k2, where n1 n2 population of species 1 and 2 respectively, k1 k2 equilibrium population size for each species in absence of other, t time r1 r2 per capita rate of increase of species 1 and 2 respectively. Then comes alpha 1 2 is a competition coefficient representing the inhibitory effect species 2 on species 1. A21 is a competition coefficient signifying the inhibitory of species 2 by species 1. For any point in between the two isoclines, species 1 is still below its isocline and increases, while species 2 is above its isocline and decreases. The joint movement of the two populations is down to the right, so species 2 is driven to extinction and species 1 increase until it reaches carrying capacity K. The open circle at this point represent a stable equilibrium. In this scenario, that means species 1 is winning. Species 1 always outcompetes species 2 and is referred to as the competitive exclusion of species 2 by species 1. In the case of second, species 2 win. How? The second scenario is the opposite of the first. The isocline of species 2 is above and to the right of the isocline of species 1. Except that the joint trajectory of the two populations when starting in between, the isocline is up and to the left. In this case, species 2 always outcompetes species 1 and species 1 is competitively excluded by species 2. Plot the isocline for the two species together to examine population trajectories. Competitive exclusion with an unstable equilibrium. An unstable equilibrium, only one of the two species survive. Which one survive depend on initial population densities. Plot the isoclines for two species together to examine population trajectories. Now we are leading on to evolution topic. So far we mentioned some important aspects related to ecology and environmental science. Next is about evolution. Abiogenesis. Louis Pasteur's test of spontaneous generation. Reddy's experiment. These are some basic ideas. You know what this means. Chemical evolution. Apparent and Haldane's theory, Haldane soup. simple molecules, complex molecules, building blocks, aggregates, cassavates, and microspheres. Then protocells, primitive stage before formation of true cell. Protocells plus nucleic acid gives self-replicating system that finally lead to a cell. Chemical evolution, Urey and Miller experiment. In 1953, Stanley Miller and Harold Urey tested the Oparin-Haldane hypothesis using the conditions of prebiotic Earth. Electrodes spark discharge gases, methane, NH3, H2 water, and hydrogen. Water out, condenser, water droplets, water containing organic compounds, liquid water in trap, boiling water. Theories of evolution, Lamarckism, new needs. The changes in the environment factor like light, temperature, medium, food, air, or migration creates new need for the organism. To fulfill these needs, living organisms have to decide which resultant changes in habitat and habitat of the organism. Lamarck's giraffe, short-necked ancestor, stretches to reach leaves and stretches to reach higher leaves, long-necked descendant. Use and disuse of organs. Continuous use of certain organs makes them active and strong, while disuse makes them inactive, reduced, and ultimately it disappears. Inheritance of acquired characters. Lamarck believed that every generation have acquired new characters which are transmitted to new generation. After few generations, new species is formed. But this is of historical importance only. Then Darwinism, rapid modification. The reproductive potential of living organisms is much more than actual survival, like paramecian reproduction rates, limited food and space, struggle for existence, like intraspecific and interspecific competition, variation, survival of the fittest. New Darwinism. According to New Darwinism, adaptations result from multiple forces and the natural selection is one of them. The ultimate character results due to the interaction of the determinants as well as the interaction between organisms and the environment during development. Darwin's finches. They are big size changes. Evidence of evolution. Homologous organs. Those organs are similar in their basic structure or origin but differ in their looks and function. They show very light modification which lead to divergent type of evolution. HD for short. Four-limb invertebrates, leg in insects, mouth parts of insects. Homologous organs. Man, cheetah, whale, bat. Bat wing, butterfly wing, bird wing. These are analogous organs. Okay, those are the organs which differ in their basic structure or origin but are similar in their look and function. It is a convergent type of evolution. Wing of birds and wing of insects. Connective links. The living organism which possesses characters of two different group of animals or taxonomic categories. Such animals indicate transitional forms of organic evolution. Examples: organism connecting link between virus, non-living and living animals, virus, animals and plants, Euglena. Annelida and Arthropoda, Peripatus, Annelida and Mollusca, Neuplina. Bony fishes and cartilaginous fishes, Chimera, Protozoa and Porifera, Proterospongea. Reptiles and mammals, Echidna. Missing links. Missing links are organisms possessing characters of two different taxonomic categories. This is an image of an Archaeopteryx which have Ornithorhynchus, duck-billed platypus, reptiles and mammals. Wing claw, reptilian character in case of Archaeopteryx. Toothed beak, reptilian character. Airfoil wing with contour feathers, avian character. Long tail with many vertebrae, reptilian character. Natural selection, industrial melanism. Natural selection of neo-Darwinism operates through differential reproduction, which state that best adapted organisms mature earlier and leave more progeny than which are less adapted. Well adapted organisms contribute more to gene pools. Industrial melanism. Peppered moths are commonly found in England. Originally, they were light-colored moths living on the light-colored lichens that cover the trees. Their light color effectively camouflaged them from predators. Peppered moths are commonly found in England. Originally, they were light-colored moths living on the light-colored lichens which covered the trees. Their light color effectively camouflaged them from predators. During the industrial evolution, coal-powered plants emitted both sulfur dioxide and soot. The sulfur dioxide killed off much of the lichen, while the soot darkened light-colored trees and stones. Light-colored peppered moths stood out brightly against the new darkened background and were easily picked off by birds. Meanwhile, the darker colored moths lived longer and reproduced. In fact, darker peppered moths had 30% greater fitness advantage as compared with light colored moths. 90% of peppered moths were dark colored. Mode of natural selection, this is very important. Directional selection. One extreme trait against the other extreme. Long wiggly tails look like a snake and scare predators. The longer the tail, the more it looks like a snake. That's kind of advantage. So extreme traits are favored, that is directional selection. Then disruptive selection. Both extremes against moderate traits. Short tails help keep predators from catching you on the ground. Long tails are good for balance in the trees. Medium tails don't help. This is the case of disruptive selection. A middle state, that is a stabilizing selection, selection against both extremes. Moderate traits against both extremes. Short tails mess up the cat's balance. Long tails drag on the ground. Medium tails are best. That is the stabilizing selection. So extreme characters are favored in directional selection. Both the extreme characters work against as the stabilizing one in case of disruptive selection. But stabilizing selection is one where the middle portion or the moderate traits are favored. Here we can see stabilizing selection in detail. Original population is there. Population after natural selection kind of sort of favored the extreme character. Robins typically lay 4 eggs, an example of stabilizing selection. I'm sorry, it's stabilizing selection, that means middle one is favored. Robins typically lay 4 eggs, an example of stabilizing selection. Larger clutches may result in malnourished chick, while smaller clutches may result in no viable offspring. Stabilizing selection removes extreme variance from the population and preserve intermediate types. This reduces variation and leads to maintain the status quo for a particular phenotypic character. Okay, directional selection. Extremes are favored. Light-colored peppered moths are better camouflaged against a pristine environment. Likewise, dark-colored pepper moths are better camouflaged against a sooty environment. Thus, as the industrial evolution progressed in 19th century England, the color of the moth population shifted from light to dark, an example of directional selection. Selection changes frequency of alleles in one direction. Genetic diversity is reduced. Knockout maladapted genotypes. Examples: drug resistance in tuberculosis and HIV, agriculture, artificial selection for species specific traits. Sexual selection for a trait for bright plumage. One extreme is favored. Disruptive. Both these extremes are favored. Like Original population is their population after natural selection. Disruptive selection happens in large populations with lots of pressure for the individuals to find advantage or niches as they compete with each other for food to survive and or partners to pass on their lineage. In a hypothetical population, grey and Himalayan grey and white rabbits are better able to blend with the rocky environment than white rabbits. They are really easy to be found out, so they get caught, resulting in diversifying selection. Disruptive selection examples: in an environment with deep and shallow flowers, short beak hummingbird leaves can get pollen from flower. Medium beak hummingbird dies cannot get pollen from flower. Long beak hummingbird lives to get pollen from flower. So that's it about three types. Next, pattern of evolution: convergent evolution. Convergent evolution is a process by which two species develop similar features despite not sharing a recent common ancestor. Next, we will learn with the help of next video. Thank you for listening.